Welcome everyone to BrainMap. This seminar series is co-sponsored by the P41 Funding Center for Mesoscale Mapping, housed in the Martinos Center. So I'll just hand it over to Dr. Delara Lucia, so we can introduce our speaker. Navarro de Lara. There we go, sorry.
No problem. So welcome everyone. It's my pleasure to introduce our speaker today, Victor Souza Oliveira de Souza?
Souza.
De Oliveira Souza, sorry. So Victor is the director of the Connect to Brain Laboratory and a research fellow at Aalto University in Finland. And he's developing instrumentation methods and applications for brain stimulation. So he obtained his PhD in medical physics in 2018 at the University of Sao Paulo in Brazil. And Victor has created and validated several human and preclinical multi-coil TMS devices to study network brain functions and created the first open source neuronavigation software for TMS, named Invasalius. How do you pronounce that?
Yeah.
You have to pronounce that story, which is currently used in many research groups worldwide. His experimental research focuses on understanding the human motor system. So I would like to remind our audience to please address any questions using the QA box or raise their hands at the end of the talk. So Dr. Souza, thank you very much, and the stage is yours.
Yeah, thank you very much. And it's, of course, a great pleasure to be here and show our work. I think there's a lot of things that we can discuss and interact, and it's always really good to get some new ideas and have good discussions, especially when we are developing new instrumentation and on the side of applications. For instance, that's always excellent to get a great insight. That's again, thanks a lot. And I was in the Society for Neuroscience meeting the last days. So that was a great opportunity to broaden a little bit our view on the kind of what's going on in the field of neuroscience and so on. And one thing especially caught my attention, which is basically the advances in neuroprosthetics and kind of the use of neural interfaces to actually restore function. So here in the video, we have, for example, a recent study that used an implanted device in the brain that actually transmits the information to the spinal cord, and that helps in for example, restoring the ability to walk in a paralyzed patient with spinal cord injury. So I think that's one of the big goals that we all have, is like to be able to restore function, even if it's like a psychological kind of impairment or motor impairment. So that's a big thing, let's say, for us to think about. This is a schematic representation of the device or that they have applied. So the group applied. It's a few electrodes placed on the cortical or with surgery. So it's implanted device, and also there is another implanted stimulator on the epidural electrical stimulation here, and which this connection between these two devices allowed the transmission of the signals from the brain to the spinal cord. But nobody wants to really go for surgery, right? So it's always a tricky process, and that also hinders the possibility of large-scale studies and to understand the motor system or the function in general. So there's a big need for non-invasive technologies that allows us to do the same thing, but without having to go through surgery. We all know that the motor pathways or basically how we control our muscles and movements is a very complicated process. It's really nice to study that because we can have immediate responses from the muscles, the connection to the brain, and so on. But we shouldn't ignore that there are a lot of different brain regions that are involved in this communication process, like from visual to some motor areas to the cerebellum, spinal cord, and muscles, and so on, so that we are capable of accurately reaching some object or performing daily functions. And TMS, as most of you know then, is a very good tool to investigate the motor system, for example. We can easily apply a pulse and then evoke activity from the brain that goes through the cortical spinal pathway or the cortical spinal tract, reach a muscle, and we can measure these activities or these evoked responses from the muscle through EMG or directly from the brain with electroencephalography, and create sort of maps of the spread of activity or kind of the relationship between certain areas in the brain. We can also use TMS to map the representation of muscles at the cortical level. We can apply pulses in different brain regions and record the muscle response, creating sort of a map of representation and identify which locations are associated with a specific movement or a specific task. This is very useful when, for example, doing surgical mapping. When a surgeon needs to remove a tumor, he or she needs to identify which areas we need to actually save or we need to avoid damaging, because that will kind of help in the prognostics of the patient, the recovery of the patient. We also have TMS as a treatment tool if applied with repetitive pulses or some other bursts of stimulus to actually modulate activity of different areas at the long term, for example, in treating depression. How TMS is applied? We have basically here a conventional commercial system. There's a person holding a coil, presses a pedal, evokes a stimulus from the muscle that we can visualize. So here you can see the person really kind of trying to find the location where the response can be evoked. And that has a very limited accuracy in general, if you compare to kind of invasive techniques. It can be very slow. So it's very hard to manipulate manually this movement of the coil. And because of that, you usually need a very experienced person to perform this experiment. So it takes a lot of time to learn how to use it, to get all of the tricks on how to position the coil, and so on. And so it's a skilled activity, let's say, a skilled task. And also a very important thing is that it's sort of blind to the connectivity of the brain in most applications, and we lose a lot of functional and anatomical information when applying TMS as it is applied now. So one big question is like, how can we drastically improve this technique? And well, first of all, we can only do that through the development of new technologies, and in a way that this technology would allow us to accurately and precisely deliver the stimulus, basically at a millimeter scale, would be already a huge improvement. It should be fast because we need to engage with different, with the dynamic oscillation in the brain that goes, basically the neuronal activation goes at the millisecond level. It needs to be automated because the brain is changing its activation very fast. So there's a lot of brain states and so on. So if we want to engage with the brain states, we cannot do it by hand and looking at the response. It needs to be automated. And we should incorporate some more priors to the process. For example, anatomical and functional connectivity measures. So if we have a conventional TMS, what we have been doing in our group is augment all of the technologies related to TMS and the targeting of the brain by using, instead of only TMS, we use multi-locus TMS. So that's one key development in our group, by using multiple coils to manipulate the stimulus. Instead of targeting manually, we can replace the manual with a robot to automate the targeting process. And instead of just having a navigation, we can actually use functional and anatomical information to enhance or augment our targeting capabilities. Well, this is the main result of the whole presentation. That's the key thing we need to we achieved. So we have a robot that is placing a stimulator on a desired target on the brain. On the left, we have a navigation system that is showing there in real time some anatomical connections inferred from diffusion MRI. If the subject moves the head, the robot compensates those movements and can automatically reposition the stimulator at that desired location. And when we reach the desired location, the mapping can be started automatically and done without moving the coil, for instance, or the coil set. multi-locus TMS, that's the technology that we developed for very fast and accurate change and manipulation of the stimulus on the cortical surface. And that is achieved by combining multiple coils in a single stimulator. So basically we have at each of these layers one coil shape, and each one will induce a specific stimulus in the brain, and we can combine those fields into a desired target. To control those, we have to develop then custom power electronics, and each of these components were designed in our group. And of course, here we have different IGBT bridges to control the current waveform, and some powering capacitors and resistors. And they're all enclosured in this big, almost like a refrigerator size stimulator connected to the coils through the cables. So just a quick overview on the electronics, which is based on the H-bridge design with the coil here in the center. And we have 5 of these modules in our latest device, which is a 5-channel stimulator. And through closing and opening the IGBTs in the circuit, we can manipulate the current waveform as we wish. Here in this case, we have a pulse current that is flowing basically through different states of the IGBTs. And then with that, we induce a certain electric field profile in the brain over time. With the coils, as I mentioned before, how the MTMS works, we have several coils, and one coil induces a specific shape, electric field shape in the brain. Here we have a figure-of-8 coil that induces this sort of blob. And then we have a circular coil that induces a ring-shaped electric field. And here a more complicated coil shape, like a clover leaf. And if we apply a stimulus at the same time in all of these coils, we can achieve a certain electric field shape in the brain that we desire. If we just change the currents without moving the stimulator, we can actually move the focus of that stimulus to a different location. And that can be done within milliseconds and just by changing the currents in the stimulator. So this sub-millisecond or millisecond level changes allows us to perform paired pulse stimulus, for example, at different locations without moving the stimulator. And also RTMS protocols at multiple locations and so on. And that is desired, for example, if you want to target different nodes of a certain local network in the brain. Here in this demonstration, just note that these changes are amplified, so that's not the true range that we achieve with the current instrumentation. It's just for illustration purpose. To coils, the process or the pipeline that we use, we have the brain. As good physicists, we always simplify everything as a sphere. So the brain in this case is a sphere, is modeled as a sphere, and then we use a conventional or a commercial coil, figure-of-8 coil, and generate a bunch of targets in the brain that we want to reproduce. We create a sort of a geometry of the coil that we want for it to have, for example, a planar or a curved surface. And then through that, we compute the minimum energy current distributions on these coil planes that would reproduce those targets that we generated. And we use that using an optimization process. And in the end, we decompose these values in components, and we can discretize those current density into isolines or into lines that would then become our coil winding paths. With this coil set, we can basically have a very high resolution targeting and change the focus or the shape of the electric field at the cortical surface. We can, for instance, move the field and the orientation of the field in a specific cortical region from this location to a more medial location, for instance. And that can be done within a diameter region with the current setup. We can expand that a little bit by compromising some other parameters that we follow. This can be done, this targeting can be done using a realistic head model as a reference. So we can basically click in the brain which location we want the field to be high and which location we want the field to be low, and create this sort of anatomically related electric field map. And this is also done using an optimization process with some constraints. If we then change electronically without moving the coil set, millimeter by millimeter along the central sulcus or the hand knob in this case here, we can do this high resolution motor mapping and have, for instance, which are the areas of highest muscle response with very, very good granular resolution. If we can do that electronically without moving the system, we can also start thinking about making a closed loop system in which we have the coils, and then we give a pulse and some algorithm that reads that response and decides where should I stimulate next. So this is sort of an adaptive stimulation guiding, which would then allow us just to put the coils there, press a button, map, stimulates, and in the end it will give us what is the optimal location and optimal orientation for that response, to actually get a muscle response. Just an example here how this algorithm works. We give a pulse, get a certain MEP amplitude. Then we give a pulse in a different location, which is marked here by this vertical line. And then I get another muscle response. And I keep doing that iteratively until the algorithm finds what is the optimal cortical location to get the response. So this is mapping along a line. Okay. The same algorithm can be expanded for single trial EEG responses. Of course, this is much more complicated due to the low signal to noise ratio. So we need, in principle, to select a certain combination of peaks to take to monitor, and we can also find what is the optimal orientation, for example, to maximize the amplitude of this peak to peak. Of this certain peak to peak that we define using single trials of EEG. And in the whole automation process, this is a bit not linked yet. It's not linked anymore to those, but is a step towards automation of extraction of parameters. We can also annotate latencies and peak-to-peak amplitudes. Well, the peak-to-peak amplitude is very easy to annotate, but the latency is pretty tricky, and it has been done manually for a long time. But we could use then some sort of pre-trained network based on sort of labeled MEPs and latencies to make this process very fast and automatic. So we have done that by developing this algorithm named DELMAP, based on a big data set of MEPs that we have annotated in our group. With the MTMS technology, we tried to kind of create some algorithms for automating the hotspot searching process. Also kind of by changing the properties of the waveform, we can extract neuronal membrane properties, because we can change at the microsecond scale the duration of the waveform that we give. And that allows us to kind of look at the neuronal depolarization time constants. And by applying the stimulus in different locations and different orientations at the millisecond interval, we can, with paired pulse, probe some inhibitory and excitatory mechanisms and how they work at sort of a network level. This at a local network level, and let's not think about like a whole brain network level. And some of the studies we have published on basically the technology on how to develop the coils, on how to kind of manipulate those parameters. But this has all been done with basically we go in there and holding the coil by hand or fixing it close to the location that we want. Now, our next step was to basically jump to the full automation, even on the positioning of the stimulator. And for that, we developed a robotic-guided system that would really help us in the positioning of the stimulator and automate further the targeting process. Of course, it's really hard to kind of develop your own research tools in closed platforms, commercial software, and we have been using the Invesalhos software, which I have been helping in the development since 2008 or so, for a long time already. And this software is a neuroimaging processing software. It was initially developed for computer tomography, and then we expanded that for TMS, navigation, and MRI applications. It's free, open source. Anyone can download and play around with that. It's hosted in GitHub. And since this year, we have had some support from Google, which has helped us expanding the MRI support on that. One of the new features that we implemented there was the capability to have markerless navigation, because for some applications, and we take, for instance, like TMS EEG, sometimes it's really hard to place the head tracker, and because we need to rely on a very fixed position on the head, and sometimes the cap comes over it, and then if it moves, it's quite problematic. One workaround for that is just not to have at all a head tracker, and we can do it by facial recognition of certain landmarks using pre-trained also networks that would identify the certain landmarks in the head, on the face, and then with that track the head positioning. So the head position here is marked as this yellow, is taken as this yellow marker here. And of course, it fluctuates depending on changes in facial expression. So this is, of course, a limitation, and depending on the angle that the person is looking at the camera. But that is already an improvement if you're doing rest experiments, short, where the person doesn't move much or doesn't change, smile or cry. Yeah.
What's your sampling frequency there?
I don't know. Usually it's the sampling frequency of the camera here.
60 hertz, that's pretty high.
That's about 60 hertz probably.
Yeah.
So yeah, that should be around that. Yeah, so this helps us then in removing the head tracker. But now looking inside the brain, we can use also functional and anatomical information. So from different MRI sequences, it's really useful to get that kind of information. So far, what has been done in the field is basically follow the grayscale maps with certain, and then you need to know, okay, here's the central sulcus, here is the hand knob, here is our DLPFC, more or less, and so on. But what if we would know the connectivity of that region with other regions, or how does that region connect to other areas of the brain? And a very useful tool for that is tractography, or diffusion, which is extracted from diffusion imaging. And with the very fast algorithm developed by our colleague Dogu Baranaydogan, we can compute this tract in real time, given a seed in the brain, and use that to infer our targets in the TMS during the navigation. Another possibility is to use functional MRI overlays, and that was the latest addition to Invesalhos. And for TMS navigation with tractography, that's how it looks like. You bring the coil, we sort of infer where the coil is projected into the cortical surface, and from that region we compute the streamlines and where does it connect to other regions in the brain. Of course, linearly projecting the coil is not optimal, so we also need to know, have an estimate at least of the dosage and the electric field in real time. So we have been using also fast computations of the electric field to guide the stimulation. So this is really new. I recorded this video before coming to US. We are fine-tuning the speed and optimizing some of the processes still, but that's how it looks. It currently looks, and that's a work of a PhD student, Anna.
Question. Or was the volume conductor modeling that you showed?
So do you mind repeating the question for the Zoom audience?
Okay, yeah. The question was, what is the volume conductor used for this? So this, in this case, it's a single shell kind of extracted from the MRI. It's a single shell BAM model.
Realistic.
Realistic.
Okay.
Yes, realistic. Yeah. Then also, we shouldn't be only focused on human applications, because we have limited capability of studying disease models, and of course the preclinical studies open a whole new different level for us to kind of manipulate and create the different disease models. And we should kind of also have a side view, or kind of view on the preclinical side, but that is extremely hard to do. So TMS on small animals, rats and mice, for example, is extremely difficult to perform these experiments. Usually the brain is, not usually, but the brain is very small compared to the coil size. It's very difficult to keep it focal. So there are some serious technical challenges that we have been trying to address. And one possibility is to combine TMS, TMS, MRI for preclinical studies. And we have made or have developed one MTMS device that can be placed inside the MRI shielded room. And this is how it looks like. We have an MRI scanner, 9.4 Tesla, and it's in another city in Finland. It's in Kuopio. And here, this is the 9.4 Tesla scanner. We have optical fibers connecting from the control room. So here is the computer and the FPGA and all the electrical control components that are then transmitted through optical fibers to the MTMS cabinet. Here and here we have the capacitors and the H bridges, which are connected then to the coils placed inside the scanner. This is a photo of the installation there. Compared to the commercial TMS systems, which are usually placed outside the shielded room, then we have much shorter cables, and that's basically like one big help, I think. But running TMS inside MRI is crazy difficult, especially if you are in a 9.4 Tesla. This is one of the first prototypes, and basically what happens is that you give a pulse, the coil breaks. So that happens at very low intensities of stimulation. Here you can see the fracture on the coil plate. And this can pose really big risks if you are running with animals, and not to say with humans. And the reason for that is that there is a high mechanical stress that comes from the Lorentz force. And what we've done, we've tried to incorporate simulations, where we try to use simulations then to know which regions of the coil plates are under the highest stress, so that can help us in designing the coils kind of with reinforced regions, let's say, that would prevent it from breaking. And what we can see that there is a shock wave propagation when we give a pulse. I don't have the time scale here, but it's just an illustration like how it goes. So it propagates when you give the pulse, the region of stress propagates, and then probably due to the geometry of the coil, it reflects on the walls and then come back and then intersect in different regions and deforming the plate. So we need to kind of have some good strategies to minimize that. One step that we took was in using materials with different kind of elastic properties, so that we would dampen, in principle, the impact of the initial impact of that. But that's another problem, because if you try to put soft materials with good dampening properties inside MRI, you will also have high signal coming from that, and that can interfere depending on the sequence that you are using. Here we're using a fluoroelastomer rubber as a bed for the wires. That helped quite a lot, and we could reach suprathreshold stimulation intensities there. This is how the TMS coil looks like with the RF coil that was also fine-tuned to work in combination with the TMS coils there. And then we have a handle to put the animal and the coils inside the board altogether. This gives us the possibility then of this coil set specifically of changing the orientation of the stimulus without moving the transducer, because you can see here this is a kind of almost on scale diagram, and we cannot basically move the coil set inside the MRI. It's really hard to do that. So the electronic targeting here would play a very big role, because that would allow us to change the stimulation location and orientation without moving the coils. And we can basically, by changing the stimulation, record different muscle responses. Here in this case, we have recorded this outside MRI, because when we recorded this data, we were still not very well. We didn't want to expose the animals to the risk of breaking the coil and eventually and going there. So this recording part was done outside, just to demonstrate that the orientation, electronic change in the orientation actually evoke different types of responses. And inside the MRI, we did a simple check to see if that would interfere with the images. And here we did an anatomical image just to see that, okay, the MRI doesn't show any, the coils don't show any distortion, big distortion here or in the field. We can clearly see the rat brain as a phantom in this case. And we also applied interleaved pulses, TMS pulses, and recording of the MRI signal, of the free induction decay signal. And we can see that as we increase the intensity, we need a slightly higher delay between the TMS pulse and the start of the recording of the fMRI signal or the RF signal. And that basically depends a little bit on what orientation of the stimulus we are applying, because that is related to different currents in each of the coils. So that's one thing.
But that image is only anatomical.
That's only anatomical, yeah. Yes.
Distortions you see it in the function.
In the function. Okay, yeah. And then the other kind of following, basically, we extracted the data from this chart here, from this one, which shows us how much the RF signal is disrupted by the TMS pulse. So this is the normalized power spectrum for each delay between the TMS pulse and the MRI signal recording. And then here we can see, for example, at the 25% of our maximum stimulator, like at 3-4 milliseconds, we basically disrupt the recording of the MRI signal, but after that, the signal recovers pretty well. So this is just to illustrate that also it's possible to run this combination. Of course, this is an effort of a big group of people. Not all of them are listed here. It's impossible to put all of them here. And in the past several years, we have been working on this project, and with a lot of support from the funding agencies. Thank you very much, and it will be good to discuss further questions.